Yes, I would like to introduce Dr. Al George of Northwestern University. Dr. George started working with the AHC Foundation in 2012. His current research project is exploring mechanisms and the therapeutic opportunities in AHC, which will hopefully give additional clues into AHC. Dr. George.
Thank you very much, Lynn. It's a pleasure to be here. Let me just do a sound and visual check. Can everyone hear me okay? And see the slides that are on the screen right now? Great, okay. Thank you again for the opportunity to talk to you today. It's a privilege to work with the AHC Foundation, and it's been an important mission of our lab to try to understand the fundamental mechanisms underlying alternating hemiplegia of childhood, as you'll see, using a unique cellular resource that we've been able to collect from individual patients with this condition. What I'm going to do today is really present to you where I think we are in our understanding of the fundamental cause or fundamental mechanism of AHC. This is work that's, as Lynn pointed out, it's been going on for several years, and sometimes it takes several years to sort of tease out what the underlying cause or mechanisms are for a rare genetic disease. But I think we are there, or very close to being there, at least understanding some peculiarities of this condition at the cellular level. And my hope is that the information that we've learned can be important barometers or measures of success of new therapies that can be tested in this cellular platform that we've been using. So let me begin, first of all, apologizing. I know that a large number of you listening today do not have a scientific background, and some of the things I will be saying will sound like complete Greek to you. I will work really hard to explain the fundamental principles that I'm trying to present, and I apologize in advance if I lose some of you. Let's start with the most fundamental thing, which is ATP1A3, which is the name of the gene associated and causative of AHC, encodes a protein that we refer to as an ion pump. This is one way of describing the structure of that protein. ATP1A3 is one of 4 similar ion pumps that is specific for the ions sodium and potassium. This is actually the structure of ATP1A1 illustrated here. There's another way to illustrate this protein in its functional, in a functional schematic. This is the protein that we're talking about. It's actually a protein that sits in the cell's membrane. Part of it faces inside and the other part faces outside. And what this protein does is it transports these 2 ions, sodium ions, shown here as Na, it removes from the cell, and potassium, shown here as potassium as K, brings potassium into the cell. And it's powered by a metabolic source of energy called ATP, hence the term ATPase. So this is an ion pump. All cells have similar ion pumps. ATP1A3 is specific to nerve cells throughout the body. And so this is a schematic of how it works. And sometimes, although it's really important to realize that this imbalance of how it pumps sodium out for potassium in, 3 sodiums leave and only 2 potassiums come in. There's some really important physiological significance to that. If you're having trouble remembering that 3-to-2 ratio, this following animation probably will stick in your brain. I didn't do this, somebody posted this on the internet, but I'm delighted every year to show it to families who may have never seen this before. And also I always show it to medical students who get a kick out of it. But this basically shows you a more popular version of what the pump is doing. A little more boring look at that is, again, if you look at how the pump is working, it transports 3 sodium ions out from inside the cell to outside. At the same time, it moves 2 potassium ions into the cell. Because of this imbalance of movement of what are positively charged ions, there's a residual deficit of positive charge inside the cell, and that leaves a little bit of a negative charge. And so that's what's illustrated here on the right, that nerves have an electrical charge across their cell membranes, with the balance being a negative charge inside the cell. Now, the ATP1A3 is one of the two broad factors that contribute to this. I'll introduce you to the other factors that contribute to this charge distribution in a moment, which I think is really important for understanding AHC. So we do know that the ATP1A3 is important for helping to generate asymmetrical ion concentration gradients across the cell membrane, whereby sodium is concentrated outside the cell in exchange for potassium, which gets concentrated inside the cell. And these concentration gradients of sodium and potassium are vital for the function of nerves. Anything that disrupts that chemically or genetically will affect the ability of that neuron to perform its most vital function, which is to generate an electrical impulse called an action potential. I'll say more about that in a moment. What we know about AHC from genetics is there are large numbers of mutations that affect critical functional regions of the protein that are probably majority, if not all, reduce the function of the sodium and potassium pumping of this protein. We call those loss of function. Now, we've been modeling AHC using induced pluripotent stem cells. This is a technology developed within the last 20 years for which a Nobel Prize was awarded. This takes, the technology takes advantage of the fact that you can take any cell from a human body and reprogram it to be a stem cell. And a stem cell is simply a type of cell that can be then converted to any other cell in the body. So a very primitive cell that can be used to generate a heart cell, a skin cell, a neuron. And then those human neurons, if you will, can be used for a variety of scientific research purposes. We've, over the years, in collaboration with Kevin S., Kathy Swoboda, and many other researchers, have now collected a large bank of iPS cells that represent a multitude of mutations, mostly represented by the three most common AHC-associated mutations, D801N, E815K, and G947R. We have some others as well that are a little less common and may offer insight into specific aspects of the disease. I'm going to focus what I talk about today on this mutation, G947R, largely because most of the data that we've generated was associated with this particular mutation. And that's simply from historical perspectives. That was one of the first lines that we were successful in creating. The kind of research that we've been doing is really aimed at doing a couple of things. First of all, we've used this cell model to determine what is the functional defect of mutations in ATP1A3, and how do we directly prove that. Once we knew that, we wanted to turn our attention to what does that tell us about the fundamental disease mechanism? What causes AHC at a molecular and cellular level? And then this cellular platform can be used to test the effectiveness of different therapeutic modalities, including some of the ones that Josh discussed in the opening, that more of which you'll hear about, I'm sure, tomorrow. With iPS cells, we can generate two major classes of neurons. Those neurons that elicit an excitatory response, in other words, they stimulate electrical impulses in a target cell, or neurons that elicit an inhibitory response. In other words, they suppress the tendency of target neurons to fire action potentials. These are the two major types of neurons in the brain. They're not the only two, but these are the major two that we are going to wrestle with. Here's just an example. This is a microscope image of two neurons, two classes of neurons. On the left are excitatory neurons that we've identified by lighting them up with a red fluorescent protein that is only expressed in, or is driven to only express in the excitatory neurons. And on the right are inhibitory neurons that are identified by a green fluorescent protein. And these are very reliable methods that we can use now to generate these two different classes and then go to work. So I'm going to start with a summary of everything I'm going to tell you. So hopefully some of this will make sense as I go through it, but I'm just going to summarize for you what I think we know right now about the cellular and molecular mechanisms of AHC. First of all, there's a primary failure of the sodium-potassium ATPase ion pump. Instead of being able to pump sodium and potassium, it just lies there inactive. There are many reasons for it to be inactive, but that's the primary underlying functional defect in this disease. But that doesn't tell us enough. What are the consequences of impaired pumping? Immediately, it's evident that if the pump isn't working, it's unable to generate these asymmetric concentration gradients of the important ions sodium and potassium. Imagine this ion pumping as a mechanism to basically charge a battery. And that battery derives its energy from these ion gradients, high amounts of sodium outside the cell, but low amounts in, and high amounts of potassium inside the cell. That's basically the charge on the battery. If the pump fails, it's like your iPhone charger's wire is cut. You don't get a charge. And if you don't get a charge, you can't elicit electrical impulses. Now, another clue to that early on was that we noticed that the resting charge on the cell membrane was abnormal. Normally, it should be pretty negatively charged, but in the AHC neurons, it was not. It was less negatively charged, or what we would call depolarized. It should have been more negative, it wasn't. That was a direct consequence of these impaired ion gradients. And these two important properties result in an inability in some cases for neurons to actually fire their electrical impulses. And that is probably at the root of many of the signs and symptoms of AHC. Up to this point, I've told you about two important ions, sodium and potassium. But there's a third ion that's indirectly related to this story that we've recently investigated, and I think it's going to be part of the story, and that is chloride ions. And chloride is important for the maturation of neurons, particularly a particular class of neurons, the inhibitory neurons. And we think that may result in delayed maturation of neurons that could explain some aspects of the condition. At the end of the day, we think impaired excitability of neurons, impairing their ability to fire electrical impulses, is what is really responsible for attacks of weakness and maybe attacks of dystonia, depending on the balance of which type of neuron is affected. And this other more esoteric effect resulting in impaired neuron maturation may explain some elements of neurodevelopmental delay that is prevalent in AHC. So let me walk you through these major things. First of all, how did we demonstrate that the ATP1A3 mutation caused impaired ion pumping activity? We used an electrophysiological approach that allowed us to directly measure a small electrical charge that builds up on neurons when the sodium-potassium pump is active. Because there's an asymmetry in the movement of sodium ions and potassium ions, there's a little bit of a charge that occurs with every pumping cycle. When we start out these experiments, we can place the neuron in a bath that contains no potassium outside the cell. That would cause the pump to not have any substrate to activate itself. So we see no current. But if we then suddenly switch the neuron to bathing in a high potassium concentration, we see this current flowing in the cells. We call this the pump current. And this is not something we discovered. This was known to occur in all kinds of cells as a byproduct of the sodium and potassium pumping action of ATP 1A3 and ATP 1A everything, all the different ATP genes. Now, if we take the potassium away, that current goes away. And then if we repeat this in the presence of wabane, which is a specific inhibitor of ATP genes, ATP 1A3 and 1A1, et cetera, we blunt that effect. So this gives us an idea of how much pump current is in the neuron. Now, these were done, first of all, on normal, healthy neurons with no ATP 1A3 mutation. If we repeat that in a neuron that has a mutation, in this case, G947R, the height of this current is greatly diminished. And we can actually infer from the wabane experiment that we've lost a large amount of the wabane sensitive part of this current, which we attribute directly to ATP 1A3. This was the first direct evidence of impaired pump activity in the neuron in AHC. We've also made this observation for other mutations, including D801N and a couple of others. And this work was first published back in 2018 from our laboratory with work supported directly by the AHC Foundation. Now, knowing that the pump is not working in iPS cells or cultured cells was valuable, but we also wondered if, in fact, if we took a neuron from a fully developed brain, whether we would see the same thing. So another project that the AHC Foundation has funded in our lab is to create a mouse with this mutation. We have succeeded in doing that, and we're now actively characterizing the behaviors and neurological dysfunction of those mice. I'm not going to show any of that today, but I will show you evidence that if we take neurons from those animals, we see the same phenomenon as we saw in the iPS cells. So when we take it from the animals that don't have a mutation, we see a nice large pump current. G947R mice, we see a blunted pump current. This is just simply the quantification of the data. So I think this observation is real. It's not just a cell culture phenomenon. It's present also in mice. And this is an important readout. Any kind of gene therapy or gene correction that can be done in these animals, this could potentially be an important readout to show that there's been a fundamental molecular correction of the gene defect. Some would call that target engagement or something of that nature. I'm going to move on. So knowing that the pump isn't working, we wanted to evaluate whether or not we could determine if that failure of the pump was significant enough to disrupt the ion gradients in the cell. So this cartoon simply shows the cell membrane of the neuron, and the sodium-potassium pump, illustrated by this structure, is removing sodium from the cell by bringing potassium into the cell. That sets up conditions whereby sodium ions are very highly concentrated outside and very little is inside. And by contrast, potassium is depleted outside, but is concentrated inside the cell. And again, this is the battery of the neuron. If you don't have these steep gradients of potassium and sodium, the neuron can't really work properly. Well, when the pump is not working properly, you get this. You get less pumping, you get more sodium residing inside the cell, and less potassium as compared to normal. That's a problem. And what we think will happen, of course, is that that's going to fundamentally affect many things. The first thing it's going to affect is the distribution of these ions will affect the electrical potential or the charge on the membrane. Now, we had a theoretical way of evaluating that called evaluating the potassium equilibrium potential. And this is something that would be complicated to explain to a class of medical students. But the bottom line is what this information tells us is it gives us a way of back calculating the concentration gradient of potassium based on an electrical measurement. When we look at cells that do not have a mutation, the equilibrium potential is about minus 92 millivolts. And that's what we would expect. Based on that measurement, we can actually calculate what the potassium concentration inside the cell is, and that number comes out to be 127, pretty close to what was predicted. In AHC neurons, and again, these are neurons with the G947R mutation, the equilibrium potential is much less negative, minus 64. And that predicts a very different intracellular potassium concentration of only 46. So we expected it to be 127, or predicted it to be in normal neurons, but it's only 46 in the AHC neurons. This is clearly abnormal. This is going to influence many things in the cell, but one thing it's going to affect is that resting charge on the membrane. We can calculate that using a sophisticated equation based on these numbers. And our calculated resting potential for the normal neurons is around minus 60. Whereas AHC neurons, it's much less negative, minus 46. So we can actually also directly measure that. And we also did that as well. We took the neurons, we tested the voltage across the cell membrane, and this is what we see. In the case of no mutations, we get close to what we predicted, minus 59. In AHC, minus 49. There is a 10 millivolt difference in the resting membrane potential. Some of that is simply because of these disturbed ion gradients directly attributed to ATP1A3 dysfunction. But it turns out it's not the whole story. One of the other major contributor to this resting charge on the membrane are a group of ion selective channels called leak channels. And leak channels are there to help the membrane help to rebalance these ion gradients. There are sodium leak channels that allow sodium to drift into the cell at a very low level. And as sodium moves into the cell, it's undoing part of the work that the sodium potassium ATPase has done. Not all of it, just a little bit of it. And that will help to depolarize the membrane a slight bit. On the other hand, potassium leak channels are there to allow potassium to leak out, and that causes the membrane potential to go the other way, to more negative potentials or in the hyperpolarizing direction. And these are just part of the normal resting.
Join the meeting.
This is part of the normal resting physiology of a neuron. Well, in AHC, instead of a high concentration of potassium inside the cell, there's a low concentration. We estimated maybe even as low as 50 millimolar. That's going to diminish the drive for potassium to leave the cell. And that's going to reduce the contribution of potassium leak channels to the membrane potential and could very well explain a large amount of that depolarized membrane potential that we see in the AHC neurons. To prove that, or at least to gather supporting evidence, we turned to a compound called BL1279 that is known to directly activate potassium leak channels. And despite the low potassium concentration and the low driving force on potassium to leave the cell through these channels, this compound could activate and restore that. Does that work? So our hypothesis was that the leak channels are part of the problem of this depolarized resting potential in AHC neurons. So if we measured the resting membrane potential, this resting charge on the membrane in neurons with the G947R mutation, we see a resting potential of around minus 58, I think, or so in this experiment. If we add the drug or the compound, we get it back toward normal. So in this case, sorry, it was minus 54, and the drug boosts it back to minus 61. So it has a partial effect, not a complete effect, but it has an effect. So this suggests to us that a component of this depolarized resting potential is the blunted potassium ion gradient that indirectly affects potassium leak through potassium leak channels. We can partially recover that with this tool compound to prove the point. So in other words, activation of leak channels helps to correct the resting potential. Now, that's one element that we have worked on. Now, ultimately, it's how do those things impair firing of the neurons? So, as I said before, a neuron's primary job is to transmit electrical impulses down its long, drawn-out process called an axon, and then to convert that electrical energy at what is called a synapse, or a chemical connection with another neuron. Now, electrical impulses in neurons are well understood. A lot of this information comes from studies from years ago, where scientists took a very long nerve axon and stimulated it with an electrical current. And then at different physical locations downstream of the stimulus, they made electrical recordings. And what they saw was there was a phasic change in the membrane voltage that was observable at places close to the stimulus or far away from the stimulus. And this is what's called an action potential. This is the fundamental electrical unit of information that's traveling along nerves. And it's important to know that the position of these electrodes that are recording this event are spaced apart. And simply, this is the time after the electrical stimulus is given. The action potential arrives first at point A, and then later at point B. And this suggests that that electrical impulse travels along the nerve, just like in the cartoon here. Now, why does this happen? What is responsible for this sort of up and down change in the membrane potential? That is also well understood. We know that it's due to the sequential opening of our voltage-gated sodium channels. These are different from the leak channels I just described. And secondarily, opening of voltage-gated potassium channels, these guys. These open and allow sodium to flow into the cell or potassium to leave the cell. As sodium is flowing in, the membrane potential drifts up, and as potassium then the sodium channels close, then potassium channels open and the membrane potential goes back. So this is sort of an event that occurs over the course of a very short time frame, a few milliseconds or thousandth of a second. Sodium ions flow in because of the work done by ATP1A3, which causes a high amount of sodium outside the cell. So there's a lot of sodium outside that can then drive it into the cell when these sodium channels open. And similarly, it collects potassium inside the cell and gets it ready to be driven out of potassium channels once they open. Now, every time this happens, you dissipate those sodium and potassium gradients, and they have to be reconstituted by sodium potassium ATPase. What happens if the pump isn't working? There's going to be a delay or even an impairment in replenishing those ion gradients so that this mechanism for firing action potentials can be regenerated. So ATP is important for restoring basically the balance of sodium and potassium in the cell. And without that, the nerve can't regenerate an electrical impulse. So we looked at this. We looked at the ability of these neurons to fire action potentials in the laboratory. Here is a non-mutant, or what I've labeled here as a normal neuron. And what's being done here is we've stimulated the neuron with progressively larger and larger and more intense electrical stimulation. When we have no electrical stimulation, there is no action potential. If we hit the neuron with a little bit of electrical current, we can see a few spikes. If we turn up the juice a little more, we see more spikes, more and more spikes. Eventually, we get to a point where we're not seeing spikes. When we really turn it up, there's a few spikes in the beginning and then nothing. And this tells us that at certain electrical levels of electrical stimulus, the neuron fires so intensely that it ceases to fire after some period of time. So action potentials fail. This is part of normal neurophysiology. The neuron you can think of as getting exhausted. It's fired so much, it's basically dissipated the sodium and potassium gradients. The membrane potential has also gotten to a point where it's preventing the neuron from regenerating. We call this depolarization block, but the bottom line is this is a phenomenon we expect to happen. In AHC neurons, and here again, it's the G947R mutation, we see this happening at much lower levels of electrical stimulation. These neurons appear to get exhausted easier or more readily. I'll give credit, by the way, to Simon Frost for directly and somewhat indirectly suggesting this experiment that we did in our lab. So these neurons don't recover as well as normal neurons when the ATP1A3 pump activity is impaired. Some of that is directly related to the ability of the neuron to regenerate the ion gradients. The rest of it is probably due to the fact that the membrane resting charge or the resting potential is abnormal, and that predisposes the neurons to failure. It's possible that this is the fundamental reason why there's episodic weakness in your children. It also may explain dystonia. It depends on which class of neurons is predominantly affected. If we're predominantly losing excitatory neurons that are involved in movement and muscle contraction, this may explain failure to move and hemiplegia. However, if it's the inhibitory neurons that are failing predominantly, then perhaps those inhibitory neurons are unleashing unrestricted muscle contraction or potentially dystonia. That's hypothetical at this point. I think there could be more research done at the level of an animal model to try to investigate that. But this is sort of where we are right now. The last topic I want to talk about is this concept that neurons with ATP1A3 mutations may have delayed maturation. And before I get to that, I want to again go back to this sort of fundamental idea that there are these two broad classes of neurons. There are neurons that elicit excitation, or their ability to excite electrical impulses in an adjacent cell or a neighboring neuron. And then there are inhibitory neurons. And there's, at any given time, our brains are working with a particular balance of excitation and inhibition, as illustrated by this seesaw. And there is fundamental information known about excitation. In most neurons that elicit an excitation, they use the neurotransmitter glutamate. And neurons that elicit inhibition use the neurotransmitter GABA, and that's an acronym for gamma aminobutyric acid. Just call it GABA. There's a lot of information known about this balance. Normal activity reflects a particular balance of excitation and inhibition, but imbalance is thought to be a fundamental observation in conditions such as epilepsy, autism, possibly AHC. So what determines these activity of these neurons. So we, two classes of neurons, those that elicit excitation. They largely use glutamate as their neurotransmitter. They depolarize adjacent neurons, and that causes depolarization and action potential firing. Inhibitory neurons use the GABA as their neurotransmitter. That tends to hyperpolarize target membranes and suppress action potential firing. So think of this excitation as the accelerator pedal, and inhibition, inhibitory neurons, as the brake. And the brain needs a brake. It needs to temper activity at any given time. If there's too much excitation, that could mean a seizure or something else. Maybe that could partially explain dystonia. Interestingly, in early development of the nervous system, the inhibitory neurons act more like excitatory neurons. And the reason for that is the GABA neurotransmitter has particular effects on the target cell that it acts on. In early development, GABA affects those neurons differently than in later development. And so we think of GABA neurons as undergoing a maturation process during early brain development. I'm talking really early brain development. And this makes me need to explain that process of maturation and bring in a new ion into the conversation, and that ion is chloride. The maturation of GABAergic neurons involves changes in chloride ion balance in the target neuron. So in early development, neurons that contain receptors for GABA as a neurotransmitter, GABA receptors are basically conduits for chloride ions. In early development, neurons are loaded with chloride. They have a high chloride concentration inside. And because of that high chloride concentration, if you open up a pathway for chloride, like a GABA receptor, responding to the presence of GABA, chloride is going to leave the cell. Chloride, which is negatively charged, as negative charges leave the cell, it's going to make the interior of the cell less negative. That's a depolarization. Whereas in mature neurons, a new mechanism has arisen, that has allowed chloride to get out. And that causes intracellular chloride to be much lower. And now the situation is changed. GABA chloride channels, GABA receptors that are chloride channels, now allow chloride to come into the cell. That brings negative charges into the cell, which is a hyperpolarization. Completely different than the immature neuron. Whereas the immature neurons respond to GABA by depolarizing and exciting their membranes, mature neurons, which occupy a fully developed brain, the response to GABA should be hyperpolarization and a suppression of the tendency to want to fire action potentials.
Dr. George, you just got about 5 or 6 more minutes, okay?
Thank you, I'm almost done. So what we did was we tried to measure or infer what was the state of these neurons in culture. So we were able to measure the response to GABA of neurons that either had or did not have an AHC mutation. And we looked at that transition from the beginning, where there was all the cells responded to GABA in the immature way, versus when they responded in the mature way, or 100% of the cells were matured. And this is the time course of that in culture. It took about 50 days to fully mature. Those are cells without a mutation. However, the cells with G947R, there's a delay, and there's probably even a failure to reach a full degree of maturation. This is a similar phenomenon that's been observed in Fragile X syndrome. There's a suggestion of it in autism spectrum disease models. And there's a suggestion here now from our data that maybe this is also happening in AHC. This is thought to be a mechanism for neurodevelopmental delay. So with that, I just will leave you with sort of our summary, which is we've been using human induced pluripotent stem cells from patients with AHC to investigate these fundamental molecular and cellular mechanisms of AHC. Our main observations so far have told us that the mutations in ATPase1 and 3 we can show directly result in impaired sodium potassium ATPase pump activity. We've shown that to be true also in a mouse model of AHC. And there's a consequence of that. The main consequence is we have impaired ion gradients of sodium and potassium. That results in depolarized resting membrane potentials, and that impairs the ability of these neurons to fire, and indirectly, it delays their maturation. And with that, I just want to thank the great team of people in my lab, particularly Christine Simmons, who's been the primary driver of AHC research in our group, and is responsible for all of the great work with the iPS cell-derived neurons. Chris Thompson, who's provided electrophysiological expertise to this. John Mark de Keyser actually developed the mouse model I talked about. And none of this work would have been possible without some collaborative work that we started back at Vanderbilt with Kevin S. and his group, and the generous supply of biomaterials, biospecimens from Kathy Swoboda's lab and Emmanuel Rose's group in France. And certainly, again, I want to thank the AHC Foundation for their long and enduring funding of our research, and also thank Hope for Annabelle for more recent contributions to our work. And with that, I'd be happy to take a few questions. So thank you very much.
Dr. George, I'd love to jump in with a question if I can. I'm pretty excited about your work in general, and thank you so much for all you do for all of us. The insights into the GABAergic neuron development and the depolarization block are, of course, important to explore and use in our understanding of AHC. I'm especially excited to see numbers associated with wild type versus mutant pumps, including voltage at resting potential and intracellular potassium concentrations based on the Nernst and Goldman-Hodgkin Katz equations. I saw that your box and whisker plots on the voltage data are quite tight and they look statistically significant. How robust and repeatable are those experiments, and how much can we rely on those voltage and concentration differences to test for a rescue?
I think they've been reproducible in multiple lines, some lines with the same and some lines with different mutations. And so I think they are reliable. I think these are tedious measurements, but we have enough information now, I think, that we can predict the number of measurements we have to make to robustly determine statistical significance. So I would say that we are we're there at the point of showing reproducibility, and I think we can definitely assess the efficacy or effectiveness of therapeutic maneuvers.
Very exciting. Thank you.
See, there's a lot of questions in there. Is someone going to hurl them at me, or should I read them?
Lynn. Is that Lynn? You're on mute. There you go.
Sorry about that. Okay. Let's see here. Question. While the research presented focus on one mutation, are they finding that the results are similar to the list that you presented earlier?
Yeah, so we've focused on that first mutation. Again, it was one of the first lines we had, and we also had additional lines so we could reproduce the work. We have now done some of the same measurements in at least two other neurons, two other mutations. D801N shows the similar impaired pump current and depolarized resting potential. And we have a splice site mutation, which is an unusual mutation that also shows similar phenomenon. We have not yet looked at E815K, the other major mutation. That's something we are desperate to do. We've collected enough lines now that I think we can proceed and do that, but we just haven't done it yet. You're muted, Lynn.
Sorry, too much going on here. Thank you for that. Another question is, what is the electric current stimulates equivalent for a human?
Oh, that's a good question. I don't know that I can answer that. I think that when we're applying these electrical currents on individual cells, it's very dissociated from what you would need to do on a human, an intact brain, for example. And so I don't think I can answer that.
Okay. And we have quite a few questions here, and I think we are almost out of time, but let me ask one more question, and then can I send the rest of these questions to you, Dr. George, afterwards?
That would be great. I would be happy to write out answers and send them back to you.
Okay, awesome. So the last question is BL1249 a potential future treatment option?
Yeah, so BL1249 is really a chemical, and it's been used in lots of research, but it does not have drug-like properties. It would not be something you could give to somebody and have it absorbed and reach the brain. I do know that there has been an effort to make a derivative of it and to make it more drug-like. There so far hasn't been a lot of work done on that. I don't have access to that derivative at the moment, but I will reach out to those chemists to see if I can get some, or if nothing else, we could make it. But yeah, I think hypothetically, if one could activate potassium leak channels, that could be therapeutic, but I worry also that that's a mechanism shared by a lot of general anesthetics. And so it might simply induce sleep. But we know sleep does counter attacks, but we haven't really conceived of this as a potential therapeutic approach. But it is something, if we can, we would test in our mouse model. We just need to get the right chemical version of it that can have drug-like effects.
Okay, thank you. Thank you, everyone, for those great questions. I will forward them to Dr. George, and then I will get the answers back to you. Dr. George, thank you once again for your presentation. Really appreciate your time this morning.
Thank you very much.
Thank you.